So for sake of time, I think we'll get started. Welcome everyone to today's OTS webinar. We're super excited to be able to feature two of our trainee representatives to the OTS Board of Directors. Today we have Chantal Ferguson and Hassan Fakih, who will be presenting a little bit on their research and a little bit on their intentions for the board and how you may be able to get involved. And so we're just going to jump right into it because we have the two presentations today. If you have questions for our speakers, you can ask them in the Q&A or you can send them in the chat and just let us know. You can send them while the speakers are speaking, and then at the end we will moderate it. All right, so I'll pass it over to Chantal.
All right, good morning, everyone, and thank you for having me today. I'm really excited to be one of the new trainee representatives on the board of directors along with Hassan. I'm looking forward to working closely with everyone. So I'm originally from London and I grew up in the Boston area. I went to Wesleyan University for undergraduate where I studied neuroscience and had a minor in Middle Eastern studies. And then I did research in Stephen Devoto's lab on the regulation of muscular development in zebrafish. After graduating from university, I spent 2 years as a research associate in the Breast Cancer Center at MGH, where I focused on providing post-treatment care and studying treatment-related complications, such as breast cancer-related lymphedema. I started my MD Ph.D. training at UMass Medical School in 2015, and I'm currently just finishing up my 7th year, and I have 1 more year to go, which is very exciting. I did my thesis research in Anastasia Karova's lab focusing on the development of therapeutic siRNAs for neurodegenerative diseases with a particular focus on Alzheimer's disease. Clinically, I'm interested in continuing my education in radiology or interventional radiology, and I will be applying into residency programs for these this coming fall. So with that, I'll just jump right into the science. I'm not going to go through this schematic and the pathway of RNAi, but what I will point out is that small interfering RNAs are particularly short 21 to 25 base pair duplexes. And the short length of the siRNA duplexes allows for their relatively straightforward and easy synthesis in the lab setting at this point in time. The ability to synthesize siRNA in the lab introduces opportunities for the inclusion of chemical modifications, such as those to the 2' ribose group, the backbone 5' phosphate modifications, and the addition of bioconjugates to the passenger strand, which can direct tissue and cell-specific delivery of the siRNAs in vivo. One of the most compelling characteristics of siRNAs, in my opinion, is that they are what we like to call informational tools. This means that they are comprised of a separate ionophore and pharmacophore. The ionophore is made up of the chemical structure and modifications and is largely responsible for determining the pharmacokinetic and dynamic properties of the molecule, while different targeting sequences make up the pharmacophore, allowing the target sequence to be easily interchanged without impacting the overall characteristics of the siRNA itself. And this has been demonstrated so well with GalNAc conjugated siRNAs, but there is a great clinical need to identify siRNA structures that function well in the brain and can be applied to multiple neurological conditions. And it's really important to note that like other oligonucleotides, we have limited tools that allow for siRNAs to cross the blood-brain barrier. And so siRNAs have to be directly administered into the CNS, either via direct injection into the lateral ventricles or into the intrathecal space. They can also be administered directly into the parenchyma of the brain. And so keeping this in mind, it's important to understand that for an siRNA to be effective in this organ, it would have to spread throughout the entire brain after an administration into the CSF space. It would need to be retained in the CSF for long enough to allow for continued exposure and transport into the brain parenchyma. And it would also need to be efficiently taken up into cells to allow for RNA interference to occur. And so we turn to the brain itself to guide us in designing an siRNA for the CNS. What do we know about the gross anatomical and microscopic cellular structure of the organ that can clue us into its function, the pathophysiology of disease, and can aid in the development of therapeutics such as siRNAs? Now, looking at the brain, here we have a coronal slice of the brain that's stained for myelin. And on the other side of the screen, we have a T1 MRI of the same plane. And what we can already start to see are some of the different components that make up the brain. We can see the gray matter, primarily composed of cell bodies, and the white matter, such as the internal capsule, that are primarily composed of axon tracts wrapped in lipid-rich sheathing. And our ability to see the differences in brain structure with both the myelin stain and on the MRI depends on the water and lipid content of the brain, the density of the tissues, and its substructures. So while hydrophobicity in the brain allows for rapid signal transduction between cells via richly lipid-sheathed axons, we would expect a highly hydrophobic siRNA to have limited distribution away from its injection site, and a highly hydrophilic siRNA to dissipate completely away from the general fatty or lipid-rich brain tissue, both resulting in limited distribution, limited exposure, and limited cellular uptake. And so to test this, we conjugated an siRNA duplex to different bioconjugates with decreasing hydrophobicity, starting with cholesterol, followed by DHA and PCDCA. And these have a Cy3 label on them, so we can visualize them throughout the brain. And as you can see here, as we introduce less hydrophobic conjugates, we see more spread away from the injection site. However, when hydrophobicity is reduced too much with this siRNA in the middle, there is minimal retention throughout the CNS, consistent with the hypothesis that hydrophobicity defines brain retention and distribution. Now, thinking about the CSF, we know that it's produced mainly in the choroid plexus and flows through the third and fourth ventricle into the cisterna magna and the spinal cord. The CSF provides the brain with nutrients and clears many solutes from the brain, and it turns over multiple times per day, and most of it is actually reabsorbed into the venous circulation through arachnoid villi. And it can also flow into brain parenchyma via subarachnoid spaces around small arteries and veins. Via this route, there's direct exchange of CSF with brain extracellular fluid, and interstitial solutes, particularly small molecules, are often cleared through these spaces. And so we hypothesized that a single siRNA and possibly smaller ASOs might be cleared from the brain through these mechanisms, and that by increasing the size of the siRNA via valency, we might be able to prevent it from being as efficiently cleared as other small molecules. And in fact, when we tested a single mono siRNA, we observed very limited distribution throughout the brain or retention. But by linking two siRNA duplexes together, we were able to double the size of the siRNA molecule and observed unprecedented retention with spread throughout the brain, suggesting that increasing valency, thus increasing the size, enables broad CNS retention and distribution away from the injection site. Now, distribution is really useless if the thing that we're distributing doesn't have any effect. And so we really need to make sure that efficient cellular uptake is occurring so that RNAi can actually happen. And we look to previous examples in the literature showing that phosphorothioate linkages and the inclusion of an asymmetric tail increase cellular uptake. Although we also had to keep in mind that the amount of phosphorothioates would have to be balanced with the known toxicity of some of these linkages. And what we were able to see is that in cells, the increasing the number of phosphorothioates as well as the asymmetric tail did in fact lead to increased cellular uptake, which correlated well with silencing in cells. And in vivo, when we tested the same thing, we observed improving efficacy with increased phosphorothioate content. And now we're currently working more to optimize this, to try to reduce the amount of phosphorothioates while maintaining efficacy to increase our therapeutic index. And so all of this work culminated in the development of what we call the divalent siRNA chemical scaffold. And this scaffold, after a single injection into the lateral ventricles, distributed well throughout the entire brain of mice and non-human primates. And this is published, and I encourage you to check out the publication. What is missing from this, though, is that these studies were performed using intracerebroventricular injections, a single one into the lateral ventricles. But there are other delivery methods, as we discussed previously, that may be more clinically relevant. But what we don't know at this time point is how they will affect the distribution and efficacy of the divalent siRNA. When we evaluated the differences between ICV injection and IT injection, IT being intrathecal in mice, we did observe a difference in distribution of the siRNA, with the IT injection resulting in superficial cortical distribution and really not getting that same distribution into the deeper brain regions, such as the caudate and putamen. So we thought that this could be a brain size issue. The mouse brain is very, very small, particularly compared to the human brain. And so not only is the size difference, but the CSF flow, the blood perfusion, and really the fluid dynamics of the brain are quite different between the species. And so we turned to other animal models to try to evaluate the effect of route of administration on distribution. And to do this, we started with the sheep brain, which is a little larger and more readily affects the dynamics that are occurring in a human brain. And so in sheep, we started by comparing, once again, an ICV injection to an IT injection where we threaded a catheter up to the cisterna magna. And again, these siRNAs are labeled in pink. And so all of this pink that you see here on the gross anatomical sections show delivery of the siRNA to these regions. And I think we can appreciate that we're getting preferential distribution to the cortical gray matter, with some distribution to the deeper brain regions, and not any obvious distribution to the white matter seen here. And this is validated when we quantify the guide strand accumulation in these regions. And overall, these two methods of delivery did result in quite similar distribution of the siRNA throughout the brain. We also evaluated the use of an implanted reservoir device that allows for repeated injections into the lateral ventricles without the need for anesthesia or additional surgical procedures. And we were able to sort of simulate a loading dose type paradigm. And I think we can appreciate again that there is pink, so siRNA throughout most of the cortical areas of the brain, with maybe some increased distribution to the caudate and putamen and the hippocampus and thalamus. And this was also confirmed using guide strand accumulation. And again, we see pretty similar distribution in the cortical areas, but an increase in the deeper brain regions, including the caudate, putamen, hippocampus, and thalamus. Finally, we did a direct intraparenchymal injection into the striatum, and you can see here that the siRNA stays pretty locally when you go directly into the parenchyma, and there is some distribution along the injection tract. We did quantify this, and the quantification was consistent with what we observed in the gross anatomical images. We were also able to look at silencing effect in these experiments. And while this should be taken with a grain of salt here, because this is just 48 hours post-injection, we already are able to see really potent silencing across the board, particularly with these three administrations into the CSF. What I want to point out, which we think is interesting, is that the silencing in the cortex doesn't seem to improve significantly between the single ICV injection and our loading dose paradigm. But what we do see is an improvement in silencing in some of the deeper cortical areas, suggesting that perhaps by saturating cortical delivery and silencing, we may be able to direct additional injections and additional doses into these deeper brain regions. And so taken together, we can start to postulate that the use of an intraparenchymal injection might be great for treating local disease such as a cancer, whereas injections into the CSF, perhaps using loading doses, may be more useful in treating diseases that affect deeper brain structures like in Huntington's disease. And so far what we've seen is that hydrophobicity defines retention and distribution, valency increases size, potentially increasing retention, phosphorothioates improve efficacy in vitro and in vivo, and that route of administration does impact siRNA distribution, but there seems to be less of an effect in larger mammals. And so moving forward, we're currently investigating how we can use hydrophobicity to further direct gray versus white matter delivery. And these large animal brains, such as sheep brains, are really useful to do so because there's such a clear distinction between gray and white matter that we don't really appreciate when looking at just the gross anatomy of mice brains. And most importantly, as I said before, we're really trying to develop a new scaffold, a new platform that can be used not just for one disease, but a number of different targets throughout the central nervous system. And so we need to determine if we can translate this chemical structure to other diseases. And one disease that we thought of was Alzheimer's disease, particularly given the huge clinical need for new therapeutics. And we started with targeting the ApoE gene. The presence of the ApoE4 allele is the most significant genetic risk factor for the development of late onset AD, which is the most common form of the disease. And there's excellent proof of concept data in Alzheimer's disease mice showing that when you remove ApoE with genetic knockouts, there is a potent reduction in amyloid pathology, making ApoE a really interesting target for silencing with RNA therapeutics. And so after designing and identifying siRNA sequences that potently silence ApoE, we synthesized it in the divalent chemical scaffold, and we tested it in the 5xFAD model of Alzheimer's disease. We treated adult mice with a single ICV injection, and we evaluated silencing and pathology at 2 weeks post-injection and 2 months post-injection. And at 2 weeks post-injection, as well as 2 months post-injection, we have potent silencing across all regions of the brain that's sustained over a long period of time. And really, there's barely any detectable mRNA at the time point that we tested. Evaluating for the effect of silencing ApoE on pathology using immunohistochemistry, we can see a pretty drastic reduction in amyloid plaque formation in the treated animals here compared to controls. This is statistically significant when quantified throughout all regions of the brain. And just a zoom showing that there really is nice reduction in cortical deposition of amyloid plaques after treating with siRNAs targeting ApoE. We also evaluated using amyloid beta 42 specific antibody, and we observed very similar results that once again was statistically significant when quantified and confirmed using ELISAs for A beta 42 in the hippocampus and cortex. So there's a lot more to this story with silencing ApoE, but I'm not going to go into it today, and the manuscript is currently in preparation, so hopefully you will see it soon. And we've been able to see here that the divalent siRNA can be applied to another target and disease. And finally, I wanted to touch on something that we're investigating in different areas of the lab, and that's the effect of the impact of modification pattern and methyl content on efficacy for the siRNA and some other chemical configurations as well. And so we increased the methyl content, and with the hypothesis that this might increase stability of the siRNA, maybe make it stick around in the brain for longer, and thus result in better efficacy and better duration of effect. We first tested the siRNA structures in cell-based models, and as you can see, there's really no difference between all these structures with a 7-point dose response. There's no difference in potency and very minimal difference in maximum silencing efficacy. We moved these in vivo and did a side-by-side comparison with a low 60 microgram single injection and evaluated 1 month post-injection. And now this is a very busy slide. There's a lot of data on here, but what I really want to point out and highlight is that we see a pattern that the increased methyl content to the far side of all of these graphs it does not perform better throughout the brain for silencing ApoE, and it actually seems to perform worse, suggesting that there's a very fine balance between stability, functionality within the brain for siRNAs. Interestingly, we also did see region-specific variations in silencing, and we have the thalamus, neocortex, and hippocampus just to show those differences. And so there's some possible effect of the pattern and the interaction of the pattern and the brain region of itself that determines how these structures function throughout the brain. One area that I want to point out is the thalamus. And it's very cool, actually, I think, that the thalamus, no matter what, across chemical modification patterns, across targets, across experiments, it performs very, very well in the thalamus consistently. And so there's something going on in the thalamus that's definitely worth pursuing further. And so we've been able to see that the siRNA can be applied to multiple CNS relevant targets. And we can see that chemical pattern impacts the widespread efficacy as well as region-specific efficacy. And we're currently investigating this on a cellular biology level, looking at the effect on risk recognition, loading, recycling, so on and so forth. But I think it's also really important to think about this on an anatomical or physiologic level. And when we look at the blood flow in the rodent brain and the human brain, we see that there's a large amount of perfusion to the cortical areas and particularly to the thalamus. Now, this makes sense because the thalamus is very active. It's required in so many different pathways in the brain. But it also, highlights the importance of this area. Interestingly, when we look at studies that show CSF flow to the thalamus, it's the reverse. So there's a lot of blood perfusion, but not very much CSF flow. And so the relationship between these fluid dynamics and siRNA delivery and efficacy is a really interesting area to investigate moving forward. And this is really important, especially in disease, because these dynamics can completely change when you have a diseased brain. So the flow of fluid changes, the ventricles open up, and this could really impact how these siRNA therapeutics work in patients who are receiving them. And so as I've returned to clinical studies this year, it's been super exciting to see the connections and opportunities that exist for the study and development of oligonucleotide therapeutics. I often find myself thinking, oh, this would be great for an siRNA. This is an awesome target. Or, I'm learning why a certain delivery method that seems great in the laboratory setting might not be feasible clinically. But there might be great tools clinically that we could integrate into our preclinical investigations. And so I'm really excited, as I touched upon, to investigate further this relationship between fluid dynamics and region-specific oligonucleotide delivery. And it's also been really fun to learn about a lot of the radiologic tools, including nuclear imaging, that have their basis in chemistry and physics and are already being used in the community to study PK and PD, as was done in this study from Ionis, that really demonstrates this integration of clinical medicine and basic science really well. And another really innovative strategy that some of my colleagues have started to implement at UMass is the use of tetanus toxoid, radioactive tetanus toxoid, to look at disease progression in SOD1 mice. Normally in wild type mice, the tetanus toxoid should ascend up the spinal cord, and you can see it here over time. But in the SOD1 mice, it doesn't do that. And this is a really cool, innovative way to look at how disease progresses over time and how it might respond to treatment. So I think it's just a super, super interesting way to integrate what we know about disease with what we need in basic science. And so these are just two of many examples of the power of integrated science and integrating clinical practice with basic science. And I'm really excited to be part of such an innovative community and be able to be part of the OTS board of directors to try to expose more people, particularly students who are still in their early learning phases, to the power of translational research and the information that we we don't always think about the perspectives that we don't necessarily have when we're isolated to a certain setting. And I feel very fortunate to have been in a lab, the Kurova lab, that really does emphasize having a lot of horizontal transmission of information and education. We're fortunate to have both chemists, biologists, pharmacologists within the same lab group. And so even though I am obviously not a chemist, being exposed to chemists and working on projects with them has been really, really useful for me in my learning and education and moving forward. And so with that, I'd like to thank everybody for being here today, as well as everybody who contributed to this work. There's a lot of us who do a lot of collaborative projects in the group, and so it really is a joint effort. So with that, I'm happy to answer any questions that anybody might have. Thank you.
Well, thank you so much, Chantal. That was amazing. I love these webinars because every time I see someone present, I want to change my entire career trajectory because it's just so cool, the work that's being done in our community. We have some really engaging questions in the chat, so I'll just share them with you. So Ronald asks, he says, great talk. If you compare the different administration routes, is there any difference in HTT reduction over time?
Yeah, so we've actually seen what's really was interesting about this. We looked at 48 hours post-injection, and then in other studies, we've looked at 1 month out from there. And we see very little difference in silencing over that time. So it's sustained effect of the silencing, basically. We currently haven't done like a side-by-side comparing ICV and IT injection in sheep, but in larger animals, it does seem that both IT and ICV injection have really nice long duration of effects throughout most regions of the brain.
Great. So then Hassan asks, are you looking to test other mods than methyl and fluoro?
Yeah, I think this is a great question. Yes, there's a lot of work going on in the lab about investigating new modifications. So unfortunately, because I'm back in my clinical studies, I'm not actively at the bench right now. But as Hassan knows, there's very talented chemists in our group, including Ken Yamada, who is in charge of a lot of these new innovations. And so he's really the go-to person for chatting about new modifications. But it is really fun and exciting to see what is being worked on.
Great. Another question here. Can you compare divalent siRNAs with gapmers? What are the pros and cons?
Yeah, so I think that's a really great question. I'm not sure I have like a great answer on the tip of my tongue. But I think really what's so beneficial about having the di-siRNA is that you do have two active siRNA duplexes that we see that are both active together and can be active separately. And so they really do provide sort of a double whammy in both ensuring high levels of cellular uptake, but also efficacious cellular uptake. And with that, they seem to be overall, the level of toxicity is very low. They're very safe. They're very well tolerated in the brains of animals. And we're really able to maximize the therapeutic index without seeing any of some of the signs of toxicity that people have been worried about with other modalities.
Great. So Laura has a question. She says, nice talk. Have you tried to measure the stability in the CSF?
Yeah, and we have, and we do see that I don't have an exact time that I can show you, but we do see that when we inject it and we wait a month, 2 months, that it sticks around in the CSF, that it is staying there and continuing to circulate. It's not, because of the modifications, both at the 5' and the 3' end, it's not really being actively degraded by endo or exonucleases, and it really does seem to be stable, providing for repetitive exposure as it circulates throughout the CSF.
Great. So we have one more question we'll take, and then we'll move on to Hassan. Obviously, it was an engaging talk because we're kind of giving you a bit of a defense. This question is from Elena, and she says, do you see any effect of adding a lipophilic ligand to a divalent siRNA?
Yeah, and this is actually a project that one of the other MD PhD students who's starting in our lab this year is working on, and really looking at the hydrophobicity and adding lipophilic versus hydrophilic conjugates to the DIS-RNA. And we do see an effect. It obviously depends on, you know, how far you go down the spectrum, how much of an effect it has. So I'm not going to give anything away further than that, but I will, you know, give a little teaser that it's coming. Keep an eye out for it.
Great. Well, thank you, Chantal, so much for sharing your work with us and a little bit about your story and how you're planning on getting involved with the OTS. So now we'll move to Hassan, who will give a completely different talk. And so we're excited for that.
Thank you.
All right. Thank you so much, Erin. Do you guys can see my screen? Yeah. I'm guessing that's a yes. Okay. So thank you everyone for being here today. I'm going to be giving a talk more of like a research journey about my career, how I transitioned from DNA nanotechnology to nucleic acid therapeutics, and what role I would like to play as part of the trainee representatives to the OTS board. So a little bit of a background about myself. I obtained my bachelor's degree in chemistry as a pre-medical student from the American University of Beirut in 2016. And then I completely ditched the medical school and I decided to pursue a PhD in chemistry and chemical biology at McGill University. And I worked in the Sleeman lab. And then I'm currently in the process of being recruited as a postdoctoral fellow to the Khabarova lab at UMass Medical School. And basically, the journey that took me from chemistry to nucleic acid therapeutics actually really started also from my undergraduate studies, where I was lucky enough to be introduced to Professor Pierre Caram, who is a really science and research advocate for undergraduate students, as well as educational outreach and sort of activities. And I was lucky enough that he provided me with a summer research internship where I worked on a project related to DNA biosensing. And basically what we developed is these polystyrene microbeads that are monodispersed and functionalized with gold nanoparticles. And then what we did is we functionalized them with DNA hairpins that are labeled with a probe. And then in the presence of the specific target or analyte, this hairpin will open and you will go from an off to an on state of fluorescent signal, which signals the presence of your analyte that you're trying to detect. And basically an overview of this project, what we did, we were able to do multiplex detection as well as making an assay that goes from start to finish in an hour and a half. And that was really my first exposure to research overall. And but it also opened my eye to something that I never expected, is that nucleic acids, outside of carrying our genes, our genetic informations, as well as coding for proteins, now we can use them for other things, such as biosensing. And then I asked the questions, well, what can nucleic acids do as well, other than that? And this actually will lead me to really ditching my pre-medical studies and pursuing a PhD in the Sleeman lab in DNA nanotechnology and drug delivery. And basically here, the reason that I decided to do this is that in the lab of Professor Hennadi Sleeman, they use DNA more for its structural context rather than its biological context. And that's mainly because DNA has many interesting properties that I listed here. And what the DNA nanotechnology field uses DNA as a building block, and they start using it as a building block to build this array of nanomaterials at the nanometer scale that range from DNA tiles to DNA cubes, different polygons, even DNA nanotubes. And really, you can even build a DNA smiling face at the nanoscale using the DNA origami technique. And this really opened up my eyes to the potential of such molecules, nucleic acids. And now, as you can see, we can use them as building blocks, as I mentioned, for nanomaterials. But also, because I was a pre-medical student before, I always had interest in doing some sort of application in healthcare. And that's why I was interested in drug delivery, which is the concept of using a drug delivery vehicle that's able to take your therapeutic, reduce its site toxicity in the body, deliver it to a specific site, and improve on its stability and efficacy. And that's basically was the basis for my PhD work or thesis, where I wanted to build drug delivery vehicles that are made from nucleic acids. And then kind of the cherry on top of all of this is that discovering that nucleic acids as well, they're not only just structural material or sensing material, but also they can be therapeutics themselves. And that was kind of the thrust of basically bringing all of these components together and all these applications to basically center my PhD on developing nucleic acid-based structures for the delivery of nucleic acid therapeutics themselves. And I'm showing you here an outline of my PhD projects where basically I tried to either optimize the carrier or the therapeutic or even control biodistribution using this material. And all of these three projects that I'm showing you actually stemmed from this methodology that was developed in the Sleeman lab that we call sequence-controlled DNA polymer conjugates. And what we do using this method, instead of building an oligonucleotide on a DNA synthesizer where we add a base as base in a stepwise manner, we discovered that we can actually add monomers of polymers and also in a sequence-controlled and a stepwise manner by exploiting the chemistry of phosphoramidites. And as you can see here, we added one hexaethylene unit, and then you can add 2 or 3, or you can add different units in a very high yielding step. In one instance where we added 12 units of this hexaethylene unit, we ended up with a DNA polymer conjugate that actually assembles into well-defined spherical structures that we call spherical nucleic acids, or SNAs for short. And what was interesting about these SNAs is they're actually made from a single component. They're automated in their production. They're made from completely biodegradable material. They're highly monodispersed. And also you have a hydrophobic core that you can use for other applications. And I was really attracted by these SNAs to use them for drug delivery. And indeed, some of the early work in the Sleeman lab where Dr. Danny Busmail showed that these SNAs are indeed monodispersed. They can go into cells readily without the use of transfection agents. They can actually encapsulate hydrophobic drugs, as well as have some nice promising biodistribution in animal models. And then Dr. Johannes Fakhouri also showed that these SNAs, when the corona is an ASO, then they can actually also silence genes as well. And then what I wanted to do first is kind of basically optimize this carrier to add a little bit more functionality to these SNAs. And the way that I decided to do it is to add some responsive characteristics. And the design that I did is basically we start with these 2 pillar strands, which are the DNA polymer conjugates, as I showed you before. And then we basically hybridize this bridge strand to it in a partially complementary manner using the color codes shown here. And then once you assemble it in the presence of a therapeutic, what you would end up with, hopefully, is a responsive spherical nucleic acid that has a corona that looks like this. And your cargo now is partially bound to the outside of the spherical nucleic acid. And the way that this is responsive is basically we rely on a technique called strand displacement, where in the presence of trigger strands that are fully complementary to these pillar strands, these trigger strands will invade your structure, and then it will lead to the production of the fully complementary and more stable product, releasing your therapeutic or your cargo so it can be effective. And obviously, when we add so much modifications to our original design, we needed to make sure that we are still forming spherical structures. And we confirmed this by dynamic light scattering, where our fully assembled responsive SNA has a diameter of around 30 nanometers, and its fully dispersity is pretty good. And we also confirmed that the structure is remaining as a sphere using atomic force microscopy. And then after a series of experiments, I'm just showing you here the most important one, is that once you functionalize these SNAs with an ASO against the luciferase mRNA, we only see gene silencing in cells that were pre-transfected with the trigger strands, and in their absence, we don't see any silencing activity, indicating that we can kind of control this activity using this prototype responsive structure. However, moving forward, while working on this project, we realized by doing some literature research back in the day is that actually our SNAs and other SNAs and most of actually nucleic acid-based material, we only rely on using phosphorothioate backbone modifications, as Chantal explained, they're easy to make, they increase uptake and stability. But being exposed to my first OTS meeting, I realized that there's a lot of expertise happening where basically there's a huge array of chemical modifications. You can modify the backbone, the base, the sugar, which can really improve on your stability as well as your activity. And that's what we wanted to do. We wanted to take these SNAs to the next step because we really weren't satisfied with around 40% silencing activity. And this kind of led us to collaborate with Professor Masad Dama, who's one of the OTS community members. And we decided to collaborate with him to start introducing these FANA modifications to our SNAs in a gapmer fashion shown here. And the reason that we chose FANA is because it's a relatively new modification that showed to be highly stable and nuclease resistant. It has RNase H activity for your therapeutic, meaning your gene silencing is catalytic. And also it improves binding towards your target. So what we decided to do is to take our DNA polymer conjugate and modify this portion with the 5H5 FANA gapmer pattern. And then we started testing it in cells immediately. And what we found is that the first important takeaway is that you need to have a cleavable spacer in between your DNA polymer conjugate and the therapeutic, because when we emitted this cleavable portion, we didn't see full activity in comparison to the ASO by itself. But once we introduced a 4-nucleotide PO spacer, we saw that we were able to recover the full activity that's comparable to the ASO by itself. And then if you make that spacer non-cleavable, you see that you lose that activity. So that was one of the first takeaways that we found. And then also what we did a dose titration comparing the SNA and the ASO had at different concentrations. And we saw that the SNA is actually more potent at a lower dose compared to the ASO, meaning that the SNA is in somehow aiding uptake and activity in vitro. Then we also showcased that these SNAs are modular in their design by simply changing the sequence of your target. You can start targeting different genes. So on the left, we target a survivin gene, which is an endogenous gene that aids cancer cell survival. So if you knock it down, you will lead cancer cells to die. And that's basically what we saw here, that the SNA induced the most cancer cell death. And as well, on the right side, we targeted ApoB gene, which also the SNA showed by qPCR to be the one that silenced the most. And last but not least, for this project, kind of like our most important result is that everything that I showed you before is in vitro studies using transfection agents. But when we tested it without transfection agents over 72-hour time point, what we were able to see is that the SNAs were able to induce the most gene silencing without the use of transfection agents in comparison to the ASO or the non-cleavable version SNA. So that was it for SNAs. And then what we wanted to do next is we wanted to change gears a little bit from SNAs and look closely at conjugates themselves. And if we go back to sequence control polymers, the method that I discussed with you before, if we switch up the monomers that we're adding to this particular design where we produce a dendritic moiety, we're able actually to show that this dendritic moiety has completely different functionality actually. And another OTS trainee representative, Dr. Ali Rakwa, actually studied the properties of this dendritic moiety. And what she showed is that it has nanomolar affinity to albumin. It enhances the stability to nuclease degradation tremendously, as well as the therapeutic activity of the therapy. It's actually so then the question is, how does this behave in vivo? Because everything that we've tested so far is using in vivo, in vitro. So what I wanted to do is to see how does this behave in vivo, because everything changes. Now you're injecting in mice in circulation where you have abundancy of albumin in the blood, and we wanted to see how will that impact therapeutic efficacy. And to do that, we decided to collaborate with the Hovarova lab at UMass Medical School to conduct these experiments. And then what we decided to do is to conjugate this moiety to a fully chemically stabilized siRNA. And we wanted to compare this conjugate to another conjugate that has been pre-validated in the Hovarova lab, which is the DCA conjugate. And then the first experiment we did actually is to see if we are still binding albumin in vivo. And to do that, what we did is we inject our molecules in using a tail vein injection in mice. And then we do SEC protein profiling, where basically we see that our conjugate is always eluting at the same retention time as albumin, indicating that we are still binding albumin in vivo, just like as we did in vitro. Following that, we wanted to test the gene silencing activity in vivo as well. So now the siRNA therapeutic is targeted towards Huntington. And then after 1 week of injection using a sub-Q injection, we're able to see that our conjugates actually has comparable gene silencing activity in multiple organs, similar to that of DCA. Particularly, we see that at the injection site of the dorsal skin, we have increased activity as well. So we were very excited that this was our first in vivo experiment where we showed that this dendritic conjugate is active, is able to do gene silencing, and the future work of this dendritic moiety is actually still ongoing, where we would like to investigate even further why do we have increased local activity in the local injection site, as well as we wanted to study biodistribution in tumor mouse models, because it's been reported in the literature that albumin has increased distribution to tumor sites as well. And this basically kind of comes full circle, where I'll be going back to UMass Medical School to basically further pursue these studies on the dendritic moiety and continue this project, as well as focus my postdoctoral projects on basically developing new conjugates and DNA nanostructures that are able to control the PK/PD of siRNA therapeutics, as well as improve extrahepatic activity, as well as delivery. So coming as a conclusion, based on my story, as I showed you, that a lot of networking and educational outreach and support of research and trainees actually really came along together. All of these incidents to basically led me to where I am to do all this research that I just showed you. And being as a trainee representative, that's basically what I would like to do moving forward in the next 2 years, where I would like to get involved in education and outreach activities, specifically in high schools and universities in the MENA region, which is where I come from. And also what I would like to do is to help an increase in networking events and facilitate collaborative efforts, because that was also crucial for me to do all the in vivo studies that I did. Because if I didn't meet a lot of people from the OTS communities at such conferences and discuss with previous Hovarova lab members, I wouldn't have be able to go there and do these experiments. So I think that's a very important thing that I want to focus on moving forward. And also, I wanted to remind all the trainees who are here today that we are your representatives, all 4 of us. And we always want to represent your wishes, interests, or even issues that are raised by you to the board, especially that we have a voice on the board. So please reach out to us anytime in any method you like. And last but not least, kind of moving forward, looking to the future, as a young researcher, what I would like to do is to basically fuse the naturally synergistic fields of DNA nanotechnology and nucleic acid therapeutics, because we're both using the same material, but a little bit for different goals. And I feel that if we are able to improve and bring the expertise from DNA nanotechnology to OTS and vice versa, that would be very beneficial for both fields. So I think that's it for me. Obviously, I would like to thank everyone who has helped me over the past few years, especially people who collaborated with me during my PhD work, the DAMA group, the Hovarova group, and the Sleeman group, as well as you and the OTS community, and of course, the funding agencies. And I'll be happy to answer any of your questions.
Great. Thanks so much, Hassan. Your work is so cool, and you really have such a diverse experience of what you have been involved in. To kind of get things off, I was wondering if you could kind of speak to any tips or advice for someone who is kind of researching in one field and has their eye on another specialty within oligonucleotide therapeutics and might be able to kind of make the jump. How did you set up those networks and that?
Honestly, I know it sounds easy, and it's easier said than done, but really network, don't be scared. Talk to people, you know, during conferences. If you see someone who gave a talk that's really interesting to you and something you want to get into, like, go talk to them, say like, oh, I think your work is cool. I think I want to apply your expertise to my projects. What do you recommend? How can I do this? It's really important, and people are nicer than you think. So people are always excited to collaborate, and it's really nice. I also would like to highlight as well that if you want to get involved with learning a new technique or a new expertise from other field, that don't be also afraid to like apply for awards, for mobility awards, either from your universities or even communities. I'm pretty sure the OTS would like to support some things like this, as well as other communities as well. So that would be my advice. Go talk to them. Don't be shy. People like that.
That's awesome. I know you especially have been very effective at networking during COVID and being able to set up yourself some opportunities to work kind of shorter term in different labs. Do you have any advice, especially for networking during COVID and during kind of virtual conferences, things like that?
Yeah, I rely heavily on using direct message tools, and I'm not really scared of like spamming people. I know people get busy, but don't, yeah, like if you already have a collaboration set up, for example, like when I was at my first stint at UMass, when I went for an exchange, that's when COVID hit. So I was there for a month and a half, and I had to leave. But if you're in such a predicament, like don't be afraid to like message the people you're working with, like, hey, can you do this experiment? I'll help you set it up, I'll help you plan it. Do everything you can from your side, and like don't be afraid to message them. Like sometimes messages and emails flew by. It's not that they don't want to do it, it's just people get busy by nature. So yeah, that's kind of what I would advise to do. Also, I know that there's a lot of people who actually participated in like kind of virtual exchange programs where you don't really need to go there. We can, for example, set up a collaboration where you can make samples in your own lab and ship them to that lab, and they can perform the studies for you. So that's also very helpful, I think.
Great. Yeah, that's great advice. So we have some questions now in the chat. Ronald says, nice overview. Can you use nanotechnology to deliver nucleic acid therapeutics to specific cell types rather than the whole brain or brain regions?
Very nice question. And that's kind of been the dream, I think, in the DNA nanotechnology field. We know that we can produce a lot of different structures that have either different lipid moieties or no moieties, or just different in size or shape, pointy edges or smooth edges. And I think the parameters of size, shape, functionality, modifications, all of that can play a huge impact on where these structures go. I think it is possible. I don't think it has been done yet in a very systematic, high throughput manner. And that's kind of what I would like to do. But I think it's possible. I don't think it's been done yet. That's the short answer.
Great.
Yeah.
So Orly says, Hassan, what do you think is the #1 challenge in applying DNA nano to oligotherapeutics, or a few challenges? Thanks for the very nice talk, as always.
I think the main challenge that I tried to highlight before is that, especially back in 2018-19, when I was looking at all these DNA nanostructures that people report their applications in vivo and stuff like that, is that we really don't use a lot of chemical modifications. Stability for our structures is very important. And I think once you use your whole structure is made of nucleic acids, then now you're not just risking the degradation of your therapeutic, but also your structure. Now it's going to function as not a structure as you want it to be. So I think that's one of the main drawbacks that I felt are in the field. We're not really using this huge, fast chemical modification toolbox into our DNA nanotechnology like field. And the other thing would be also in the same line of what I'm saying, is that also we need to make sure that structures are held together. Maybe you make them stable in terms of nucleus degradation, but maybe the buffer conditions are also important. Maybe once you in your test tube they're stable, but once you inject them in the blood, they're not structures anymore. You don't have enough magnesium or enough sodium ions, for example. So also making the structures themselves stable and retain their structure, which retains their functionality, is also important. And I think that's kind of where the setback was in the DNA nanotechnology field. I think for sure there's people moving more towards that now, implementing different modifications. But yeah, I think that was one of the first challenges.
Great. So a follow-up question to that is, how do you measure that the structures are stable under physiological conditions?
It's a very good question. And I think there's a lot of people in the DNA nanotech field who have developed techniques in the lab to kind of study this. I think that there's no really direct way to kind of like fish out, for example, a DNA nanocube from the liver and see if it's still a cube. But there are some techniques and experiments that are crucial that people should do, I think, before going in vivo. Like you can, for example, incubate them in cell culture media and serum, and then look how do they look on gel, how do they look on AFM after this treatment, how do they look on DLS, and you try to gather all these different proofs that okay, your structure is still staying together, and then maybe hopefully try to do that in vivo as well. And I guess they're all kind of indirect, but doing a multiple of these techniques together will kind of support your claim that your structure is still stable and together.
Great. Well, thank you so much, Hassan. Thank you again, Chantal. You both gave us really wonderful presentations and very exciting work. So I just want to wrap things up today by giving you a bit of a teaser for the next OTS webinar. So in 2 weeks, we will, on March 10th, we'll feature Dr. Alex Garanto, who is an assistant professor in the Department of Pediatrics and the Department of Human Genetics at Rabound University Medical Center. The title of his webinar is Antisense Oligonucleotide-Based Therapeutics, or Therapies rather, for Inherited Retinal Diseases from Mutation to Clinical Trial. So he'll be sharing that with us. Then the following week, we have another OTS trainee representative spotlight. And then throughout the year, we'll be continuing to spotlight trainees in our series and then also feature other speakers. Currently, we are looking for a speaker for April 7th. So if you liked what you saw and you want to have an opportunity to tell your story, please get in touch. You can contact either Ronald or I directly or through the OTS info email. And so since it is 12, like I said, Ronald and I just want to thank everyone for your participation and also thank again Hassan and Chantal for their wonderful contributions to today's webinar.
Thank you.
Thanks.
Great. Okay, so that was excellent.